Hey everybody, this is Mr. Nixon representing here on the broadcast for today. I think my volume's a little low, so let me work on that just for a second. We're going to be talking about chapter 4 and kind of getting into that tonight. Probably split this into two separate videos for you guys. I think we're going to split it up into two parts. The first part, I want to kind of give an introduction to cells and talk about transport, how things get across the cell membrane, how do things get into the cell and then get outside of the cell, and then do a second video right after that, kind of talking about the organelles inside of a cell. And I think what I'm going to do is do something a little different than what we normally do, which I think is something that I should do more often, which is bring up these class notes, and you can kind of see me down in the corner. And don't worry if things kind of freeze up for a minute. That's just because the Google Plus is reading my Google Drive and pulling up this PowerPoint and sticking it in here. So just give it just a moment and it'll get everything underway. And as you can see, probably by now, you can see the notes and you can probably see me. And I kind of want to start down here on slide 6. We're going to kind of work our way through. Let me just give you a brief introduction to cells. You know, this is talking about cells for the sake of learning them for anatomy and physiology. So something you got to keep in mind is that cells, or the study of cells, cytology, is extremely important to anybody who expects to pass anatomy and physiology. You really need to understand how a cell works. You really need to understand what a cell is. And even if you somehow memorize your way through this chapter, which I'd probably be guilty of doing the same thing, you want to try to slowly apply the concepts from this thing as you go through your anatomy and physiology courses. Some of you are taking a whole semester of nothing but anatomy. Some of you are taking an entire semester of physiology. Some of you are playing a half and half game. You take a session or semester or quarter of anatomy and physiology combined, and then you do another chunk and then another chunk. But either way, whichever way you're doing this thing, you really need to understand how cells work, because you're made out of cells. I mean, you're made out of trillions of cells. And if you could think of a cell as a living Lego block, and the fact that you're made up of trillions of these Lego blocks that are alive, that do numerous different things to make you work. It's kind of like looking at an entire country, and every person in that country is a living cell. And it takes every person in that country to make that country great. So cells in your body are the same way. If something's wrong inside of you, then it's probably something wrong with some cells. Like when you get a cold, that cold virus doesn't just jump in your throat and say, you know, time to get down and party like it's 1999. No, that cold virus is going to find its way inside of some cells and it's going to wreak havoc. And then what makes matters even worse is your immune system is going to respond, and it's going to respond on a cellular level. And you're going to see things that your cells are going to do to respond to that invader, to that pathogen. And you're going to unfortunately feel the wrath of that invasion and that resistance on a sign and symptom level, which is what you and I can personally feel. So I kind of want to start off talking about cells by looking at this chart. And if you look at this chart, and if we increase it just a little bit, let's start at the bottom here so you can see sizes. Okay, the smallest thing that we generally discuss here in anatomy and physiology is an atom, unless of course we're talking about the subatomic particles of an atom, like the electrons, the protons, and the neutrons. Unless we get into that, generally in anatomy and physiology, you don't go any smaller than an atom. But if you take two or more atoms and you stick them together, you can wind up with a small molecule. In this case, the example they have here of a small molecule is an amino acid. Now, please don't get this twisted. This thing over here is not an amino acid. The thing over here to the right is an amino acid. It has a stereotypical group of carbon, nitrogen, oxygen, hydrogen, and then it's got this R group over here just kind of chilling. And this R group is going to basically be, whatever it is that makes that amino acid unique. And you can see the R group, it'll be over on the side somewhere. But this is just representing a small molecule, some amino acids. Each one of those little dots are representing an atom of a particular element. So there's like a carbon here and a carbon there, and probably oxygen there and oxygen there, so forth and so on. So you stick all those atoms together, you make a molecule. Then you can make really big molecules like a macromolecule. This of course would be a protein. Remember, anytime you have over 199 amino acids attached together via peptide bonds, you make a protein. So here's this protein. He's a pretty large molecule. And then you can take these macromolecules and put them together, combine them, and when you combine these macromolecules together, you can make something like a ribosome, which is a very, very small organelle inside of a cell. Now notice, we have not reached the cellular level yet. Everything that I'm blabbering about is smaller than a cell. Like this thing is inside of a cell. Like when you go to class inside of a college or high school or elementary school building, whichever level you're in, when you go inside of your school building, you would be like this ribosome compared to that big giant building. So you have ribosomes, which is a type of organelle. We'll talk about that in our second session. And then you have a virus. Now, something I want to pause on just for a minute is look at the size of a virus relative to one of the organelles inside of your cell. Because if you go further up, okay, a virus is less than 100 nanometers, which is pretty daggum small. Once you get up way more than 100 nanometers, we're up to a micrometer. Once you get just above a micrometer, you get to a mitochondrion, which is another organelle inside of a cell. It's the organelle that's responsible for producing ATP, producing energy for your cell, or most of the ATP anyway, because it's making ATP through cellular respiration, also known as aerobic respiration or aerobic metabolism, depending on who you're talking to. So this mitochondrion, another organelle inside your cell, is well over 1 micrometer long, and that's way larger than a virus. We are still inside of a cell. We have not reached the size of a cell yet. So you can see how small viruses are and how they get inside your cells. They're so tiny. Then once you go up from mitochondrion, as you're approaching 10 micrometers, you actually get up to the size of a red blood cell. And most of your bacteria are going to be between the size of a mitochondrion and an actual red blood cell. This is the first time we finally see a cell. I mean, seriously, this is the first time we actually get a chance to see a cell in its relative size compared to everything else, which is nuts, because all beforehand, everything we've seen here is deep inside of a cell, way smaller than a cell. I mean, itty bitty tiny to a cell. I mean, when you're looking at a large macromolecule inside of a cell, that's no different from using a satellite in space and looking at a human being standing in the middle of a large city. All right, that's how small this thing is. But now we see the bacteria. Most bacteria are somewhere in between the size of a mitochondrion, which is an organelle inside of a cell, and a red blood cell itself. Now, most of our plant and animal cells, however, wind up being an average of about 30 micrometers in width. So somewhere in between this gap, we have most animal and plant cells, and as we continue to go up, then we have this cell just decide he's just going to buck the system. We have a human oocyte, which is a big fancy way of saying a woman's egg cell. And so we have a female woman's egg cell, which is absolutely enormous compared to the rest of these cells, because right here is 100 micrometers, And so we are actually approaching a millimeter when we start going up this thing. Now, to you, you might say, man, a millimeter is teeny tiny, but we're talking about a cell here. We're talking about what makes you. We're talking about the ability to be able to see something that small, but it being that large. Because remember, every time a woman ovulates, she is ovulating one of these things. So there's something this large traveling through her fallopian tubes to try to meet up with some sperm cells. As we continue to go up from millimeters, we go up to centimeters, and as we start approaching, actually, once we get past this, we start approaching 0.1 meters here, which suggests that we're at decimeters. But here's an ostrich egg, so you know that's pretty daggum big, because it's 2.54 centimeters for every inch. So as we're going up this thing, we've gone to several inches long. And if you've never seen an ostrich egg, you literally, unless you have hands my big, you're not holding an ostrich egg in one hand. An ostrich egg is pretty big. It's like, to me, holding an ostrich egg is kind of similar to holding a softball. I mean, it's an ostrich egg is a little bigger than a softball in some ways, but the one end, the rounder end of the egg, is like holding a softball. So you continue up from the ostrich egg, and look what you got here. You got two different cells that are enormous. Yes, you're reading this correct. You have muscle cells and you have nerve cells that are way larger than 0.1 meters. Now I want you to think about this. 0.1 meters, okay? A meter is 100 centimeters. So 0.1 meters is like 10 centimeters. Now remember the conversion that I just gave you. 10 centimeters. It takes 2.54 centimeters to make an inch. If you do that math, you're talking around somewhere between 3 or 4 inches long, okay? So now we're going from 10 centimeters long to approaching almost a meter, and that's how long some of your muscle cells and your nerve cells are. They are that long. We're talking about some of your nerve cells and muscle cells are going to go anywhere from inches to feet. That's how long some of these things are going to go. So you can't generalize cells. You can't say, oh, I've seen one cell, I've seen them all. No, you can't do the cell. You have to pay close attention to the type of cell that you're talking about. Where is that cell located and what is its function? Because cells are very different. And if I've never told you this before, there's something you've got to understand in anatomy and physiology, and that is that structure defines function. Structure defines function. Look at how something is shaped. Look at how something has been sized, and you'll be able to figure out a lot about what that cell is supposed to do. So when you look at this, you have to realize, oh man, you know, these cells are huge. They're probably huge because they work to move the actual body, or they function on a greater, more visible role. Whereas the human oocyte, it kind of makes you wonder why that cell is so large as compared to some other cells, because red blood cells, I can go ahead and tell you, you never want your red blood cell to be the size of a human oocyte, because a human oocyte does not have far to travel. That's why it can be so big. It is being released into its own private pipeline to encounter sperm cells and hopefully merge and turn into a zygote and implant itself on the uterine wall, and and have baby colors to be picking out for a nursery or something later on. But red blood cells, you don't have to worry about them being too big. You want them to be really small, because red blood cells have to travel through really small blood vessels like capillaries. And so you never want red blood cells to be too big, because then they'd wind up having a traffic jam in a small blood vessel in your brain. The blood vessel could burst, and yeah, I mean, you can tell from the way I'm talking about it, that doesn't sound too good. So you wouldn't want that. So remember, structure defines function. So let's move on. Let's talk about one more thing before I start getting into how cells transport things. And if you don't mind, I'm going to get a sip of something here. It's not coffee, not water. It's actually lemonade. Well, it's actually raspberry peach lemonade. Just made a little bit of that just for the sake of it. All right. So let's see. We're going to zoom. Well, we don't have to zoom back out. Let's not and say we did. So what we're looking at now is, we'll zoom back out of that just a little bit. What we're looking at now is you should have an image of this. This is what we call a composite cell. In other words, it's not, everything you see here does not exist in a real cell. Okay, so you're not going to see a cell that looks exactly like this in your body. If you see a cell that looks exactly like this in your body, dude, you got some screwed up cells. I'm just going to go ahead and tell you that now. You should never see a cell with everything that you see in this picture, ever. Like, for example, over here, you've got a phloem and you've got cilia. You know, you shouldn't really see any cells in your body with flagella. The only time really we see cells in our body with flagella is when you're looking at a man and he's got sperm cells, because sperm cells have flagella. If a woman finds cells in her body with flagella, well, then we kind of probably can assume what those cells are. That's if they're in the reproductive tract. But if they're somewhere else, then that's when we're going to start freaking out and we're going to get you quarantined, because we shouldn't see anything floating around in you other than sperm cells with flagella. We've got cilia over there. Not all cells have cilia. Those red blood cells I was telling you about, they don't have cilia or flagella. Not all cells have all of these organelles. I mean, not all of them have lysosomes. Not all of them have ribosomes. Not all of them have nuclei. Not all of them have centrosomes. Not all of them have proteasomes. You know, you've got to be careful. You cannot generalize a cell. This image is simply showing you, okay, this is what we could find in a cell. If we took about 10 random cells in your body and lined them up, then we would look inside of these cells, and these are some of the things that we would possibly find in some of those cells. But this doesn't mean that you're going to find everything on this picture inside of a cell. So if you see a cell and you're like freaking out because you're saying, oh man, you know, I thought that dude said that I was going to find a mitochondrion and I was going to find some centrosomes. No. You may look at numerous cells in the body and never find either one, because it depends on the cell, especially if you look at red blood cells. Because if you look inside of a red blood cell, a mature erythrocyte, a mature red blood cell, you're not going to find any mitochondria, you're not going to find a nucleus, you're not going to find ribosomes. So, you know, cuckoo for you. There's a lot of stuff in there that you're not going to see. So now I want to try to get into how cell membranes work. I know you're like, well, where's this going? I mean, he was talking about composite cells and this composite picture, and then he was talking about what you don't find in a cell, and now all of a sudden he's jumping to a cell membrane. Well, I'm doing that because the cell membrane, as far as the structure of the cell and every other organelle inside of the cell, well, when you talk about them, it's a lot easier just to spend time talking about the cell membrane, then come back in totally different later on and talk about the actual organelles. The organelles are going to function differently than the cell membrane, and I'll explain what I'm talking about now. The cell membrane is kind of like your skin. You know, it is a protective barrier, but it's more complex than your skin, because your cell membranes on a cell are what we call semi-permeable, or you can say selectively permeable. It depends on the textbook, depends on the teacher, depends on the research, depends. But bottom line is, cell membranes on a cell are selectively permeable or selectively permeable. Let's just go with that. In other words, they are specific about what they're going to allow in the cell and what they're not going to allow in the cell. It's kind of like going to a VIP party. If you don't have a RSVP, if you don't have a reservation for that party, they're not letting you in. Me and some friends were going out one night several months ago, and it was a while back because it was really cold, and we were looking for a restaurant to go to, and we went to the first restaurant, and the first restaurant they were like, it's going to be a 45-minute wait. We were like, 45-minute wait, and then that's not counting them finally getting to you and taking your order and fixing your food. So we said, nah, we'll just walk up the street up here and find something else. So we get up the street and we see this restaurant that we have been passing. All of us have been, passing this restaurant for the years that we've lived in the city. And we said, hey, let's just roll up in here and try it out. We looked in there and there were like 2 or 3 tables empty. So we got to the door and the guy said, you got a reservation? We said, no. And he said, I'm sorry, this restaurant is actually a reservation-only restaurant. He said, he said, I wish I could give you guys a table because things are kind of running kind of slow tonight, and we're afraid that someone's going to wind up canceling the reservation, and then we're going to have these magical tables sitting here. And he said, but at the same time, if I let you guys in and these people show up, then we're out of dough. So we understood, but we came to realize, okay, this restaurant is an invitation reservation. It's a reservation only type place. That's how a cell membrane works. If you don't make a reservation, if you don't have an invitation, if you're not on the VIP list, then you don't get inside of the cell. Now, if you watched the chemistry video that we were talking on last time, you saw where I was talking about how the cell membrane is made out of a phospholipid bilayer, which basically makes it a double layer of fat, and the fact that chemicals like steroids don't need to ask permission to enter into the cell. Chemicals like steroids, they pass right across the cell membrane and go directly into the cell, whether the cell wants it to or not. Whether you administered the steroid to that person for the purpose of targeting that particular cell is besides the point. It's a steroid, fat and fat mix. So because steroids fall into the lipid family, and the cell membrane of a cell falls into the lipid family, stuff passes right on through and goes where it wants to go. So let's talk a little bit more about the cell membrane here, and we'll get you guys out of here. Let's see here. Wait for it to come up. So the first thing that I kind of want to talk about here about the cell membrane is a few key functions that are listed here: physical barrier, selective permeability, electrochemical gradients, and communication. One of the first things that you notice up there is it's a physical barrier, and it's a very flexible boundary. It allows the cell to be able to move if that cell is the type of cell that it does move. Some of the cells in your body do not move. They do not transport from one place to another. That cell is stationary. It is glued, literally glued into a space, just like the bricks in a wall, and it has no intention on leaving. If that cell is leaving, it's probably because you tore it from its wall, or that cell has died and it's being shed. So you want that physical barrier. It keeps things that are supposed to be inside the cell inside, and keep things that are on the outside of the cell. The next thing is the selective permeability, which basically allows the cell to be able to pick and choose most of what comes in and out of the cell. We're actually going to talk about that selective permeability a little more in depth today. And what you'll see is you'll actually see where cells can allow certain ions. Remember what an ion is from the chemistry chapter? Said an ion is nothing more than an atom with a charge on it. You'll see where it can allow specific ions in. You'll see where it can allow certain molecules in, but it doesn't allow other things in. It provides an electrochemical gradient. In other words, it can actually create an action potential, which is a low-grade electric current that you can measure in millivolts. And yeah, no, I mean seriously, it really your cells, certain cells can generate an electrical current. It's not enough for you to like become the Shocker, or Electro, and jump up in the Spider-Man movie and go electrocuting folks. So don't think you're gonna wake up looking like Jamie Foxx tomorrow just because you go stick your finger in something and try to juice into your or whatever. It's not going to work that way. Your cells generate these electrical currents so that they can communicate with one another and communicate with other cells. And then, of course, you have communication, which I know I just talked about the action potential, but communication in this sense is talking about the fact that it has certain receptors that respond to certain key stimuli. I'll give you an example. When two nerve cells are talking to one another, let's say you got two neurons or nerve cells in your brain that are talking to one another. Neuron number one, the presynaptic cell, wants to talk to neuron number two, which is the postsynaptic cell. And that presynaptic cell, neuron number one, will release chemicals known as neurotransmitters, which you've probably heard of. You've heard of acetylcholine and dopamine and things like that. Okay, those are neurotransmitters. So what happens is neuron number one releases those neurotransmitters, and when those neurotransmitters bind to a receptor on neuron number two, it stimulates neuron number two to respond and do what it needs to do. So let's move down and let's talk about how things get in and out of this cell membrane. I'll go ahead and tell you right now, every method of getting things in and out of a cell membrane rise and fall on two different things. There's passive transport and active transport. Passive transport is essentially just when you use a method to get things in and out of the cell without using any energy. It's free. All right. That's why one of these days I want to make a lot of passive income. I think passive income is awesome because passive income is money that I make without doing anything. I have a few, I try to look at methods of passive income. Can't say I'm really the master of it. I am certainly no Warren Buffett, but if I knew back in the day what I know now, I would have hooked up a whole lot of streams of revenue coming from passive income, because passive income is you stick your money somewhere and you walk away, and that investment winds up making money for you without you having to do any work. That's passive income. So passive transport is transporting where a cell does not have to use any energy to get things in and out of the cell at all. Now, how's it going to do this? Well, usually there's two types of ways. There's diffusion and there's osmosis. Diffusion is easy. Diffusion is just when you got a lot of something on one side of the cell membrane, and that whole lot of something wants to move to the other side of the cell membrane where there's not much of it. So basically, a more definition type phrase for that is diffusion is when you have a high concentration of a substance that moves to a low concentration of a substance using a simple membrane, which that semi-permeable membrane is, of course, the cell membrane that we're talking about. So you've got this semi-permeable membrane, which is the cell membrane, and you got a high concentration of calcium ions outside of the cell. And so the minute you open the door, the minute you open a channel on the cell membrane, that high concentration of calcium outside the cell will automatically flood its way through that calcium channel into the cell, because there was a low concentration inside the cell, there was a high concentration outside the cell, and so things like to move from a low concentration to they like to move from a place of high concentration to a place of low concentration, and they create what's called a concentration gradient, which is just from high to low. Think of it like a rappel line. If you're riding on one of those grapples or or one of those rappel lines. What used to be used by the military and was never seen as something fun, we do it as for sport now. Everybody's going to the mountains and riding rappel lines. I have to tell you guys the story one day of when my harness on the rappel line snapped, and I was hanging on for dear life the whole way down that thing, coming down a mountain into a small valley. That was an experience and a half. That was crazy. But the whole way you're coming down off that mountain and you're going down to the valley, okay, that's a gradient. You're going from one concentration that's really high to a point that's very low. So you had a high concentration of calcium outside the cell, and that high concentration of calcium begins to flood into the cell where it's actually very low. Osmosis is the second form of passive transport that we typically talk about in anatomy and physiology. Osmosis is just like diffusion, except it uses water. So generally, water follows salt. Water follows certain solutes, but it really follows salt. And wherever you have certain solutes or salt, water is going to go in that direction, kind of like a slug. When people want to kill slugs in their garden, oftentimes they'll take salt and just shake that salt all around the garden, especially on their garden path. And the reason why is because when that salt gets onto a slug, the water in the slug's body will go through osmosis. The water in that slug's body will leave the body and go into the salt, due to the fact that the water is thinking, hey, with so many solutes over there, that must mean that that's where I need to go. And so water will tend to leave one spot and will go to another. That's why, excuse me, I'm sorry, my eyes are itching there. They've been a little irritated lately. But that's why people die from dehydration when they're lost out to sea and they're just floating there in the water. You would think that they wouldn't die from dehydration because they're surrounded by all that water, but that water is salt water. So you're surrounded by all that salt water, and your body, the water in you, is fresh water. So you can kind of see what's about to happen. So you've got two different methods of passive transport. You've got diffusion, you've got osmosis. Now, I need to show you a really important concept, and it's something you don't want to forget. It's really what happens when you start talking about osmosis and tonicity or tonicity. Tonicity is the ability of a solution to change the volume or pressure of the cell by osmosis, which is not good sometimes. And what that means is, okay, remember what I said about osmosis? Water likes to follow solutes. Now, if you're like, what's a solute? Well, okay, a solute is something that's dissolvable in a solvent. Well, okay, so I just got something that probably messed you up. Let me back up, because I said these were cell basics. So let me explain something. 3 words, all right? So solute, solvent, and solution. All right? Solute, solvent, and solution. First one, a solute is something that dissolves in a solvent. What's a solvent? Something that can dissolve a solute. I mean, no, seriously, that's about as deep as we need to get. A solute is just something that dissolves in a solvent, and a solvent is anything that can, dissolve a solute. And when you mix the two of them together, you make a solution. So when someone tells you this is a solution of something, then the first thing that should come to your mind is, wow, if that's a solution, then there must be some solutes that have been dissolved into a solvent, and this solution is the mixture of those things. That's what you're looking at. So a solvent, I'll give you an example. My lemonade. Remember I told you we made some raspberry peach lemonade. Well, what we did was, we made a simple syrup, We made a reduction, a syrup reduction out of some fresh raspberries, some peaches that we got from a peach orchard, and some lemon juice that we had there. And we added some sugar along with it, and we boiled it down to make a simple syrup reduction, and then poured it over some ice and water and a little bit of extra sugar and made the lemonade. Bam, you got it. For some of you guys, you might put a little Moscato in there or something. I don't know, whatever floats your boat. Okay, we'll try to keep this PG. So, um, so if you looked in this cup, like here, I'll turn it a little bit there. Okay, I don't want to spill this on my laptop. I don't know if you can see inside there, but because I've drank a lot of it. But you can't see any sugar in here. See, like I hold the cup up and I stick my finger in, you're going to notice that there's no sugar on my finger. Okay, the sugar is dissolved in here. The sugar granules would be the solutes, and the water that went into this would be the solvent. And if you take the sugar and the syrup and the ice and all that and mix it all together, it made a solution. So oftentimes when we're looking at cells in your body, the cells in your body are surrounded by what we call interstitial fluid, which is the fluid in your tissues. Well, humorously enough, the fluid in your tissues happen to be solutions. So, you're looking at a solution. There's all types of different things dissolved in the water in your tissues. So we're looking at this red blood cell, and he would definitely be suspended in some type of solution. We call it blood. And a lot of your blood is made out of water. Actually, somewhere between 50 and 60% of your blood is nothing but H2O, nothing but water. So in an isotonic solution, remember what we said a solution was? It's when you take a solvent and dissolve some solutes in it. So we got this solution, and it's an isotonic solution. In other words, both the cytosol, which is the liquid inside the cell, and the solution, which in this case happens to be the blood, with same relative concentration of solutes. In other words, you've got just as much salt going in your red blood cells as you have going out. This is perfect, because if I got the same concentration of solutes going into my red blood cell as I do solutes coming out of my red blood cell, then that's going to keep a balance of how much water goes in and out. And so this solution is perfect for my cells. It's isotonic. That means same amount of stuff going in and out. I don't have to worry about my cells dying. All my cells look normal. Hooray. But if I mess that up, if I turn that isotonic solution into a hypotonic solution, hypo meaning below, all right? If I turn this into a hypotonic solution, then the solution has a lower concentration of solutes than the number of solutes inside the cell. So there's less salt outside of the cell than there is salt inside of the cell. This will be bad, because water likes to go where the most solutes are. All right, that's osmosis. When water moves to where the most solutes happen to be, because water is not very smart. It thinks that because there's a high concentration of solutes somewhere, that there must be a low concentration of water, and that it is currently in the high concentration area. Water's not bright enough to see through that fallacy. So water will then flood inside of the cell. It'll leave the blood and it'll go into the cell and cause the cell to swell and expand and rupture and blow up, kind of like a Mortal Kombat fatality. That's a hypotonic solution. And the last one is a hypertonic solution. And in a hypertonic solution, what winds up happening is you put this cell in a solution where the solution has a much higher concentration of solutes than inside the cell. Well, because there's more salt and more solutes outside of the cell, all the water inside the cell leaves the cell and goes outside, thinking to itself, holy moly, look at all those solutes. There must not be enough water out there. Let me leave this itty bitty cell and go jump out there in the vast quantities of water and solutes that are outside of the cell. And so the cell starts to lose all of its fluid, it starts to lose all of its shape, and then it winds up shrinking. And when it shrinks, then it'll probably die. We're almost at the point where we're going to stop. There's one last thing I want to talk about, and that's active transport, because I've talked about passive transport and all that stuff. Oh, by the way, that hypertonic solution, that hypotonic solution, that isotonic solution, all of that was under passive transport. There was no energy being used to make any of that stuff happen. That freaky stuff was happening on its own. No energy, no batteries included, no energy required, no kind of mechanisms to make it happen. But in a active transport, you got to use energy, which means that if you're going to use energy, you're going to have some kind of mechanism that's going to be able to get things in and out. And generally, we see most active transport that we talk about in anatomy and physiology in two ways. We either use a pump or we use vesicles. And if you use a pump, you have some kind of thing on the membrane made out of some type of protein that's literally pumping certain items into the cell and certain items out of the cell, where with a vesicle of a cell, what you're actually doing is you're taking a piece of the cell, wrapping it around an object, and then pulling it in or pulling it out of that cell. So let's go to this slide here, and you can see a type of pump. If you look closely, here's an erythrocyte. This is a red blood cell. And you can see the active transport protein that they're talking about. And red blood cells tend to have a special pump, an ion pump, that if we zoom in here a little bit, this ion pump actually pumps calcium. So if we move over just a tad bit, you'll see where calcium is floating around inside the cell in its cytosol, which is the fluid inside of a cell. And the calcium winds up getting pumped using energy. Remember, we have to pay. We got to pay to play. You don't just get water for free if you live in the city. Unfortunately, you got to pay a water bill. So here, the cell is paying a power bill by paying for this ATP, and it uses this ATP to help move the calcium out of the cytosol or cytoplasm of the cell, and then outside of the cell into the interstitial fluid which surrounds the cell. And this pump requires ATP, it requires currency, it requires energy. So that cell can't pump that calcium out if it doesn't have any energy to actually do it. Let's go to this slide here where we get a chance to look at vesicular transport, also known as, using vesicles to move things. Usually when we use a vesicle, usually what we're doing is we're taking a piece of the cellular membrane, folding it, and using it like a big giant trash bag. That's literally a vesicle. A vesicle is we snatch off a piece of the membrane of the cell, wrap it up like a big trash bag, stick something in that trash bag, and then float that thing out of the cell. And it's easy for the cell to get rid of something using vesicles because a vesicle is nothing more than a membrane-bound bubble. It's using a piece of the cell. So the cell recognizes itself, and it easily allows the vesicle to pass right on through. So two different types of transport with vesicles basically is exocytosis and endocytosis. Exocytosis is how we get things out of the cell, exo, exit, and endocytosis is how we get something in. Like endo means inner, so this is how we get things inside the cell, is endocytosis. Exocytosis is getting things outside of the cell. One last thing that I did want to talk about, and then we'll shut this puppy down, because I think I've already gone past my time that I had planned. When you look at these last two slides, they kind of compare the passive process to the active process. Like with the passive process, notice that it says it does not require expenditure of cellular energy. the substance moves into or out of a cell down its own concentration gradient. You have to remember that passive means free, okay? It doesn't use any energy. It just needs a concentration gradient. So if I have more items on this side than I do the other side, then these items through diffusion will, by the law of diffusion, will move from this area where there's a lot of this substance all the way to this area where there's very little. And then osmosis is the same way, except it uses water. And in osmosis, you have a lot of water over here, but there are a lot of solutes over there. And so through osmosis, the water thinks that because there's a lot of solutes over on this side and there's less solutes over here, the water thinks that there's less water over there. So the water over here will automatically diffuse through osmosis over to the other side. And then the last image really talks about the active process. And they do a really good job of this because basically in the active process, you have to use energy. And, you know, with the active process, let me say this, because if you ever read through this and you read through this in a textbook, it can be kind of tricky. When you talk about an active transport, oftentimes somebody will come back to you and they'll talk about a concentration gradient. And that much is true. There could be a concentration gradient, and you may want to actually get something from the outside of the cell inside the cell. The problem is that item needs to be controlled as to how much of that item you want in the cell at any given moment in time. You may not want that substance in the cell all at once. You may actually want that substance in the cell only when you need it. So if that's the case, then you want to control the flow, so you use an active transport process. Or you may use endocytosis or exocytosis to get things in and out of the cell. Another thing to keep in mind, we didn't talk about this, is a type of endocytosis is actually called phagocytosis. And in phagocytosis, it looks like the cell is eating something. You'll learn much later when you talk about the immune system and you talk about white blood cells that there are cells as part of your immune system. Excuse me. There are cells that are part of your immune system that engulf pathogens, and they do this by utilizing endocytosis. They fold part of their cell membrane around that pathogen, stick it in that membrane-bound bubble we call a vesicle, and trap it, and then engage it with some type of pathogenic destructive substance like hydrogen peroxide or or some other substance that is carrying some type of lysozymic enzyme to destroy and break down that pathogen. So that's where I'm going to stop today. I'm just going to cut it right there. We'll get started on the next video shortly, and we'll put these guys together. We'll talk about some organelles and wrap this thing up. I hope you enjoyed it. Thanks. Bye-bye.